Well, I think we can start. It's 9:30 already. So thank you very much for being last day here in BioSpain, here with us. I guess yesterday the tapas time took longer than expected maybe, but thanks for being here today. So the breakout session we're going to have today is going to be focused on manufacturing. First of all, I would like to thank AceBio for hosting this impressive BioSpain here in Seville this edition. Also to the speakers, thank you very much for being here today and sharing your experience with the audience. And for sure, the audience itself is not that easy to be here last day, so thank you very much for sitting here, and please feel free to ask any question to any of us anytime. So the session we're going to be hosting here today is going to be focused on manufacturing. You know that biologics are the new trend in biomedicine, is the future, but even more the present of medicine. And manufacturing is a key factor for the success of the product development in biologics. So here we have a quite diverse panel of different service providers that are key in all the activities related to manufacturing. Yes, I'm going to start with a brief presentation of the professionals, people here today. So here we have Luisa Moedo, she's MabScience León Site Director. Robert Subban, Senior Data Scientist from Sartorius Steady Biotech. And Martin Glentz, Principal Scientist from PAL Life Sciences. So well, let's start. I don't want to speak that much. The only thing I'm going to do is to introduce myself. I'm Elena Roba, Business Development Director of 3P Biopharmaceuticals. I guess you know 3P. We are the first and only CDMO in Spain, fully dedicated to contract manufacturing of biologics for third parties, 100% dedicated to make our customers' life a little bit easy. Okay, Luisa, so let's start with you. Luisa Moedo, she's director of my MabScience manufacturing site in León. Okay. She's graduated in chemistry by Santiago de Compostela University, also masters in environmental safety and health, masters in business administration and business development program with more than 20 years of professional experience, more than 14 of them in biotech. She has occupied different positions in production, program management, and technology transfer, and this experience has provided her with a global overview and knowledge of the biotech business. Since 2013, she's working at MabScience, first as technology transfer manager, and then she was brave enough to move to León and lead the design, construction, and startup of the biotech plant of the company in Castilla León. MabScience, I guess you all know this company, is a Spain-based company specializing in the process development and GMP manufacturing of biotechnology products, mainly monoclonal antibodies, from early phases up to the clinical and commercial stages. This company develops and produces its own products to commercialize around the world. So, Luisa, your turn.
So, good morning. Good morning, everybody. Can you hear me? So I would like to explain a little bit of our experience in MabScience to construct, build a manufacturing site. First of all, I would like to show you a video of what we do, just to put in the context in where we need to have the decision to construct or to build or not a manufacturing site, and what type of manufacturing site do we need. So we go ahead with that.
At MabScience, we develop, manufacture and market biosimilars, mainly monoclonal antibodies, using the most advanced technology and cutting-edge analytical methods in our processes, following strict international regulatory guidelines. First, an in-depth analysis of the reference product is carried out to obtain all the possible information about its structure. This is a stage in which a considerable amount of time is invested, guaranteeing the best results. In the transfection phase, once the DNA sequence that corresponds to the antibody is synthesized, it is inserted into the plasmid. The plasmid with the DNA transfects the cell, where the gene corresponding to the antibody and other genes needed for its translation are integrated randomly into the cell genome, and thereby producing the desired antibody. In this stage, called random insertion, a set of polyclonal cells is obtained. These cells produce different quantities of antibodies with minor differences. The development of the cell line phase starts. During this phase, the cells are isolated and analyzed using different techniques until only the one that produces the most similar antibody to the innovator in terms of quality remains. This is when it is frozen and the cell bank is obtained. This is the basis for the following phase, upstream process. In this phase, the vial is thawed and cultivated, first in flasks by shaking, before being put into the bioreactors. The technology used is single-use, which provides greater flexibility for production and prevents cross-contamination via the use of disposable materials. In the bioreactors, nutrients and physicochemical parameters are vital for cell growth. As such, temperature, pH or oxygenation are strictly controlled during this phase. In the bioreactors after the production phase, all remaining impurities are eliminated in the purification or downstream process using chromatography columns and filters, which by means of different resins enable all the undesired components to be discarded whilst retaining only the antibody which is the most similar to the reference product. Lastly, during the formulation phase and before filling the vials or syringes with the final product, the antibody is added to a solution containing the necessary excipients to stabilize it and make it suitable for patients. This is how mAbxience develops and manufactures biosimilars, with focus on quality throughout the whole process, to achieve that each product delivered to patients is highly similar to the reference product. We call this fingerprint-like biosimilars by design.
Hello. So that's the context in where we are. I would like to show you that video because the decision that we have to take as a company in the 2009 when we start is how can we build the facilities that we need that are useful and of course don't build or don't do too much investment at the beginning when we don't need it. So that's why we decide to move with that. That's it, to take the correct decision about which facility, the size, the type of technology that we want to use is easy or maybe not, it depends. Oops, sorry. Okay, there's something missing there, but it's okay. So the decision to take, that we have to take is easy, but maybe sometimes it's not so easy. The point is that now, with internet, with all the information that we have, there is all the information available that we need. Maybe it's too much information. The good thing is that as a company, we and every single company need to understand themselves, need to understand what type of company are, they need to know what do they need, the needs in the timeline, and then go ahead with the possibilities. Evaluate what are the possibilities that there are in the market. A few years ago, and I say a few, not a lot years ago, there were not many possibilities regarding the manufacturing of monoclonal antibodies or biotechnology manufacturing. That was stainless steel, and that's it. But since a few years ago, there are another possibilities. So there is a lot of information available. There are a lot of knowledge also available. So if it's like this, why we still do mistakes when we decide? And when I say why we is many companies, we included, why do we make mistakes when we decide what facility do we need? And when I say mistake, what I mean is maybe you construct a facility that is too big for your needs, so then you have 4 or 5 years your facility empty. That is a very huge cost for the company. Or in other cases, you have a so small facility, not enough capacity, so you have to go out to do your process, so you lose your knowledge, you lose time, it's more costly. So to take that decision is important. And what are, from our point of view, or what were our main drivers to take that decision and to do it in the best way? First, identify and define the topic, collect the information, identify the alternatives, weigh the evidence, and decide. That's the typical steps in any process, in any project that you want to do, okay? But the point here is that, again, knowing your strategy as company, knowing where do you want to be in 2 years, in 3 years, in 5 years, knowing and be honest with yourself, with your possibilities, with your plans, that's the main driver, to do the correct questions and to get the correct answers, to take a good decision. So before to start to do anything, there are a few decisions that we need to take as a company. And the first one is, do we want to do it at home, in-house, or do we want to externalize? That's the first question that we need to answer. In our case, the answer was, yes, we want to do it in-house. So, okay, first question answer. Do we want to invest in a new facility, or do we have another facility that we can reuse or use for our purposes? And that's the third question. Do we want to have the knowledge in-house, or do you want to go to a CMO to do it? There is, as I told you, there is not a good or a bad answer. It depends on the company, and it depends on the moment. And that's why it's very important to analyze what do you want, what is your company, what are the strategic steps of your company in the coming years to go in the right direction or not. So first, to collect information, there are two main points, the internal information and the external information. Internal information is define what are the products that you are going to manufacturing. Are you thinking in manufacturing always the same type of products, or do you want to jump and do different products? If you are going to have same type of products, how many? 3, 5, 1? If 1, maybe it's not interesting to invest in a facility. If you are going to have 6 or 7 or 5 products that have a similar way of production, it's worth it to have your own facility or not. Depend also on the markets and other things. So that's the first. Define and know what are the pipeline that you are thinking having in the future. Projects, define the projects per product. Means projects, I'm talking more about the timeline. What are you going to do? And Map Science works since the very beginning till the end. So it means that sometimes we need to start to produce just a few batches to do the clinical studies. Then we have to stop with that product, and then after the after the clinical studies finish, we can produce a commercial. So you need to know that this time is going to be a stop moment for the facility if you have only one product. If you have more than one product and you have those products in different stages, so maybe not. Maybe you will have your facility also all the time occupied because you are doing the clinical for the product A, but you are doing the commercial for the product B, but you are doing the preclinical for the product 3, whatever, okay? And of course, the markets. It's important to know which markets are you going to go, the size of the market that you are thinking about, the forecast. Of course, we don't have the crystal ball, but we do our best to predict all. When I say we say all the companies do our best to predict our future. So in that sense, it's very important to know what is the forecast market penetration that you are thinking that you are having to do, which markets are you going to produce. So all these type of things are need to, it doesn't take too many time. We think that it means that you need 3 years to take the decision. That's not true. It's just a matter of sit down, take all the information, put all together, and in 2-3 months, this information could be perfectly aligned and perfectly all together and analyzed. So it's not that you are not going to take the decision because you need to think a lot. No, it's just, in the other hand, put information there and analyze it properly and go ahead and be honest. So mainly with the forecast. Here we need to be very, very careful, because maybe we think that we are going to be in 50 countries with 70% of the penetration in the market. That's not realistic. So be honest, be realistic, and with that, go ahead with the decision and with the analysis. Then it's important to know another topics, to analyze another external points, like the competitors. How many competitors are with the same product in the market? Because it's important to know, to define this part of the markets. Which markets are we going to go? What are going to be our penetration in that market? Because if there are 3 competitors, the penetration is going to be 1. If we are going to have 6 competitors, it's different, maybe even not interesting, all these type of things, okay? If there are manufacturers available, it's not a minor topic. If there are many manufacturers available for your products, it could happen that the cost doing outside is better than doing inside. So it's something to take into account. The production capacity of other companies, the technologies that exist, the technologies available and the technologies also implemented. This is important because when we are working with the technology that is available but is not implemented, I'm not saying not to use, because we did it and we are very happy of doing it. But what we need to know that is, if it is not implemented yet, is that you are going to have some challenges, extra challenges, okay? And in these cases, the partners, I call partners, but the suppliers became partners. To develop this technology is very important together with them, to do it properly, to implement it properly, and to convince not to convince, because it's not convinced, but to show the authorities that this technology is appropriate and is good for the product, okay? And it's the best one. So when we identify the alternatives, and now in the monoclonal antibodies and in the biotech in general, I'm talking a lot. Please interrupt me to make any question if you have any question during the presentation. I forgot to tell at the beginning, because maybe at the end you don't remember the question, so please stop me at any time. Once we identify the alternatives, of course, the monoclonal antibodies and the biotech in general, but also the big molecules, are not a simple product to manufacture, let's say. They have their complications, so there are not many possibilities to manufacture them, okay? But fortunately now, we have a stainless steel possibility, that is since the very beginning it was there, and the single-use possibility. Oops, sorry. And there is the third one, that is a mix. Stainless steel for the upstream processes, for the cell culture, what are the more sensible part of the production, and single-use for downstream, that is used for buffers or for solutions that are not so affected. So that's the 3 possibilities. All of them have benefits, not an inconvenience, let's say. Stainless steel, pros, is well known and tested, was used in that business many years. The main con is that this requires a bigger investment in utilities and in the facility, and less flexibility to change to different products, from one product to the other. Single-use, pros, less initial investment and flexibility for multi-product. Cons, limited suppliers, and that's what I mean, limited suppliers that became partners, and limited maximum volume till now, okay? And the mix, pros, is well known and tested in the more sensible part of the production, that is the upstream, the cell culture, and cons is still high investment, because again, you have to put many money in the utilities and less flexibility too. In our case, you have to take two main decisions, okay? One is the type of technology finally that you are going to use. In our case, it was clear we have many products. We have different products in different stages. The single-use technology is the best one, because it makes us possible to do it by module construction and building. If I need now 200 liters, I can put 200 liters. If then I need 500, I can put 500, and then 2,000. It's not plug and play, but is, let's say, the plug and play version of the biotech manufacturing. Okay? So in our case, the decision was easy, single use, and the size of the plant, again, it depends on the markets. So the good thing of that technology that we use, that we selected, was the single use, is that we can do it by modules. So we start with 200, then we go to 2,000, 2,000. Now we have 2,000, and in the future we have the building, the room ready to put another 2,000 bioreactors, another 2,000. So it's good to be able to build as you need, so you don't have the investment stopped there and waiting for the market to arrive and all these things, okay? So the investors are not very happy when you have the manufacturing facility there, stop, okay? So then, very important to
Luisa,
the implementation.
2 minutes.
2 minutes, okay, thank you. The implementation, key points, define the suppliers, again, the partners, and involve the suppliers and the authorities in the very beginning. When you are using a very naive, a very new technology, it's important to get them involved and to, that's, let's say, they are the best assessors since the very beginning. So it's good to get them involved and ask them, what do you think about this, blah, blah, blah. So it is better to have the input since the very beginning when you can change whatever you need, okay? And then do the follow-up. And again, in that point, be very honest. And if there is any change during the process of the construction that make a significant change in the strategy, just adapt the thing and don't go ahead with something that you know that is not going to be the best option. Why 200 liters, 2,000 liter bioreactor facility in mAbScience? That's the facility in León. Because, as we talk, less investment, less initial investment, and we can grow as we need. It's pretty close to a plug and play. More flexibility for a multi-product side, that's what we have, multi-product. We have 2 commercial products right now and another 5 in our pipeline in different stages in development. And less environmental impact versus stainless steel. It seems not, because we use a lot of plastic, but in the case of the stainless steel, there is used a lot of water and mixes clean solutions. So that's clearly that the impact is much less. And that's it.
Okay, so any question for Luisa? Now is your time for that. No? Okay. So, well, now is the time for Robert. So, Robert, Mr. Suman, he has master's degree in chemical engineering from University of Lund in Sweden. He has mainly worked with R&D pharma with both product and process development, including scale-up activities. He has also experience as a project manager, managing small R&D projects as well as scale-up projects. And the combination of a scientific and engineering mindset brought him into the field of chemometrics, where he has worked as a specialist within innovation and operations in the chemical sector. Today, he works for Sartorius studying data analytics as senior data scientist, where he teaches and inspires clients to use advanced data analytics in their daily work. Eumetrics is part of Sartorius studying biotech and offers tools for data analytics based on design of experiments and multivariate technology, as well as applications for real-time monitoring and prediction. The services that this company offers includes both training and consulting in case of need. So Mr. Suban is presenting today a lecture focused on the role of data analytics in improving biomanufacturing processes. Thank you.
Thank you for that introduction. So I'm Robert from Sartorius Data Analytics, and I'm here to talk about how to assess advanced data analytics within the biopharma. So what you see here is the geometric suite, and you can see it as a platform for different kind of clients. Clients can be coming from the biopharma, it can be the chemical sector, it can be the pharma sector, food and beverage, but also other type of industrial sectors. Since I'm here about to talk about manufacturing in biopharma, I will focus my presentation on Simcha, Simcha Online, and the Control Advisor. Simcha, that is our multivariate data analysis platform. And what it basically does is converting data tables like this into highly informative plots like this. And then we do the interpretation here, and based on the pattern we see, we're building different kind of models, depending what the purpose is. So very short how it works. I think the best way to understand what multivariate is to just show a simple picture like this. Imagine that you have a variable like this progressing in time, and then another variable like this also progressing in time. And we can see that they are correlated, meaning that if one increase, the other one increase. If one decrease, the other one decrease, so to say. Now, in the multivariate world, if we use some kind of correlation pattern, and that's the whole secret behind multivariate data analysis, we can see that most of the data points fall into a quite constrained space. But there are two data points that are different, and I'm just highlighting it like this. And you don't see it here. Everything is within the upper specification limit, the lower specification limit. So the question is, why does a multivariate technology see this as a different spot compared to over here? Actually, that spot here is this spot here and this spot here. And we can clearly see that this is low and this is high. They are no longer correlated. In this case, they are negatively correlated, and that is exactly what the multivariate technology directly sees like this. So you can see this as a core thing for monitoring a process. If Simcha is our offline multivariate data analysis platform, then Simcha Online is a real-time version of it. So basically what we do, we just basically take the Simcha Online and connect it to the IT system of the production unit, and then the Simcha Online will, so to say, visualize the status of that manufacturing unit. It can be, for instance, upstream processing, it can be downstream processing, fermentation, etc. But at least it tries to visualize the process so it's easy to interpret it. And we use this kind of system for online monitoring and online prediction. The Control Advisor is an add-on to the Simcha Online or the Simcha Real-Time, and it's also best explained by just a simple picture like this. Imagine that you would like to go from A to B, and then you have the GPS for that. So what does a GPS do? It does propose some kind of route like this. And if you have a bad luck, you will see some kind of deviation along that route. It might be some kind of, I don't know, construction word or a traffic jam or whatever, so to say, but some type of deviation. In the Simcha Online world, it will look something like this. This is a starting point, and this is the end point, so to say. And we are maybe here at the current time. And then from that point, we do a forecast into the future. And suddenly we see some kind of deviation. Why is a deviation? Because it breaks this kind of specification limit here, which is the red dashed line. So, okay, we have some kind of deviation, which is the same as this kind of deviation on the GPS. But not only that, because we're doing a forecast, we can start to predict, for instance, the end quality attributes of your product. In this case, we try to predict the amount of produced material. And since we have a deviation here in the future, the amount of produced material will be low compared to historical batches like this. So this is not a good scenario. So what does the GPS then tells you if you have some kind of deviation? Well, in this case, it will propose an alternative route like this. And that is also what the Control Advisor does. Instead of going according to this route, it will propose a new route like this. And taking that route instead will increase the produced amount of material as compared to the first route, so to say. So what it does is it gives you a new recommendation on how to produce your batches. Now, since I said that I will be talking about how to utilize advanced data analytics within bioprocesses, this is normally how we see a bioprocess, and it can not only be a bioprocess, it can be a chemical process, it can be a pharma process. And what we normally see is that you have some kind of information coming from the start. It can be information about the raw materials. And then if you have that kind of information, you can add it up with the process data. We have, for instance, batch processes. They evolve in time. We have the process tags. We have the time duration for each batch. And we have a X number of batches like this. And that adds up to a quite large data matrix. And in this case, I will show you that this is an example based on 3 different scales. So you can see that there's some kind of scale-up methodology as well. And in the end, you have some kind of final condition data. It can be quality. It can be yields. You decide that, so to say. But normally, you have some kind of specification. And then we add these things together. And for instance, if we would like to predict something, and this is just a prediction model, tries to predict the VCD, so to say, viable cell density. This is just how it looks like through 3 different operational scales. And then you have the models that is explaining what is taking place, so to say. So the first part here is actually the initial condition. It comes from this data matrix here. This is the raw material characteristics, for instance, or the initial set points like initial glucose, initial lactate, etc. And then the process starts, the evolution starts, so to say, from start to end, and that is caught up in another type of coefficient bar like this. However, though, this one is quite complex because it contains all the process data, so to say. And it looks actually something like this. So if this is the start of the batch, this is the end of the batch, we see all the variables now evolve through time, so to say. And for instance, if I would like to maximize VCD, then I should be up here, so to say, in this graph, meaning that, for instance, for this variable here, which is highlighted in red, and that is the viability, it should be high throughout the batch. And then, for instance, if we look at another variable like this blue one here that I highlighted, it should be high in the beginning, and that is the glucose concentration. So there's quite a lot of information you can extract here in order to maximize or minimize, depending what your goal is, so to say. Now, this is a prediction model, tries to predict the VCD. And we can see, for instance, that we have the 3 different scales here. This is one scale, the red ones, the blue scale, and then we have the green scale here. But there is a strange thing here taking place. It says that this is a red batch. It should be actually here, the batch number 13. But it actually belongs to the green group. Then the question is, why? It should not be there, it should be up here. So we can basically just, with the Simcha drill down technology, just click and see what are the different variables contributing to this deviation. And what we see here is actually the start and the end of the batch. We can see that there's quite a lot of variation in the beginning of the batch, and then you have some kind of steady state taking place. But there are some variables like this one that is completely out of the range, and we also have something here at the beginning. So we click on this one, which is actually the pH. We're going down to the raw data, and we can see directly that the model says that the pH is far too high, and that is exactly what you see in this batch, which is that one, compared to the rest, which should be up here. So this is our, so to say, monitoring methodology, going down to the raw data through a model with a drilled down technology. We can also look at clustering of batches. In this case, we have the 3 different scales, the blue ones, the green ones, and the red ones. And we see that, for instance, for the blue group, there seems to be a group here and another group there. Why? Why is it like this? It shouldn't be like this because we try to minimize variation here as far as possible. Then we can easily just compare this cluster to the average of all the batches. And this is basically what you're going to see. Again, it's the different variables explaining what's taking place from start to end. And here we can see that the difference here between this group and all the rest is the variables that is up here and down here, not in the middle, because that is the center of gravity. We're not interested in that. And we can see that for this variable here, with this sodium trajectory, it is far too high for this group. And this trajectory here, which is osmo profile, it is far too low. So this is how we then can go down and assess the different root causes for the different kind of problems. Now, this kind of information, this is just monitoring, this is prediction, etc., can be then transferred from the Simcha offline version into the Simcha online, trying to understand what's going on in real time, so to say. And that is basically what is taking place here. So this is just a snapshot from the Simcha Online engine, which is a prediction engine and a monitoring engine. And we can see here that here I combined two different modes. One is monitoring and one is forecasting. So this is monitoring and this is forecasting. So assume that you're manufacturing your batches now, and we can see that this green batch here is a little bit strange because it has a different trajectory in the beginning. It crosses some borderlines here and here. And then we try to forecast the end of the product, so to say, the quality of the product. And this is IgG expressed here. So we can see that this green batch is forecasted to have this kind of level in the IgG. Now, compared to the blue batch, which is according to the golden trajectory, it's already up here. We do a forecast in the future, it ends up with a higher IgG. Why? Because it follows the golden trajectory, and that comes from the monitoring model. So we can now combine monitoring and forecasting, even though we're not in the end, so to say, of the batch, so to say, but we can try to forecast the end, and based on that information, we can give vital information to the operators when, for instance, when to start the next batch, when to prepare the next batch, etc. So that was very short about the geometric suite, and it's how it's used in the biopharma, and as I said, it's also used for different kind of industrial sectors. Thank you.
Is there any question for him? No? I have one. Where to start when you have tons of data? Where to start?
Yeah, that's a very good question because the thing is that people have a lot of data. The trouble is, okay, going from the data to something like this. First of all, you have to set it up, like in some kind of structured way. If that is in Excel, that's fine. I mean, that is normally how people start, so to say. But as long as you have, for instance, get it into a format like an Excel, then it's quite easy to import that to, for instance, Simcha or Simcha Online, whatever, because it communicates with different kind of formats. But the main thing is that you have to somewhat compile it together, and then after that, you can start to assess, to use this kind of analytics tool.
So now let's go for Martin Glantz. He's principal scientist at Paul Life Science, sorry. Dr. Martin Glantz, he graduated with a degree in biology and his PhD from the University of Bielefeld. He owns more than 12 years of experience in the biopharmaceutical industry, and he's principal scientist at Pal Biotech since 2017. Before that, he was senior biopurification process specialist at Pal Life Science and led a team of specialists within Germany. As member of the scientific and laboratory service at Pal Biotech, he's responsible for the scientific implementation of continuous bioprocessing in Europe. Pal Life Sciences has grown from a company producing filters for the airline industry into a global leader in high-tech filtration, separation, and purification across many markets. Pal Biotech continuously improves bioprocesses for customers at all stages of drug development. The portfolio of products and services reflects a customer-centric approach with integrated systems and enabled technologies. Dr. Glantz is presenting today process intensification using inline concentration and diafiltration on continuous bioprocessing. Thank you.
Yes, thank you for the introduction. Good morning, ladies and gentlemen. Thank you for being here, let's say. So the title today I'd like to talk about is a process intensification when applying continuous bioprocessing. That's the content, so I'd like to give you a short introduction into continuous manufacturing today, and also a short introduction into the simulating moving bed chromatography technology. And then I have a case study for you, just to show how we can intensify further our bioprocess by using an inline concentrator in here. So I talk about setup and results, process stability, and of course economics of this. So let's start with the continuous manufacturing, and the first question is, why should you consider to implement continuous manufacturing instead of having a batch process as before? So batch processes are well known, are established since many, many years. So why should you consider to move towards continuous processing? And first point would be, first point is the process economics. So it means you can improve the capital utilization, and you can, of course, reduce the operating expenses by running continuously. And also the process control. It improves the process safety and also the product quality. So you have more control about the product quality of your process and of your product as well. And for the biopharm industry, we said there are two, let's say, main arguments for the biopharmaceutical industry to go towards continuous processing, and the one is the improved capital utilization, so you can get more product out of your setup, and the other one is the improved product quality, and for sure that's not just the product, it's the safety for your patients as well. So both of these arguments should lead or can lead to continuous bioprocessing. So this is a strategy for process intensification. So you can produce more material in less time with more or less the same equipment. So it's the same way of producing, let's say, an antibody. So the same steps are applied there. You can save some raw materials, of course, and auxiliary materials as well. More compact bioprocess systems. So there are a lot of, let's say, reasons for going into continuous. I'd like to show you here how continuous performs against the batch process. So when I start this animation here, you can see here, these are some of the steps you would have into batch processing. The first bioreactor, then a cell clarification, protein A, and so on and so far. And this here is the same, but a continuous DSP process. And you can see here on these little watches. So while here, the steps are running step by step, one after each other. Here, they are running in parallel sometimes. So it means when we go into continuous, we are much, much faster with the same equipment, producing the same amount of, let's say, an antibody or product in comparison to our batch DSP. So it takes much, much longer, and therefore, we say continuous DSP might be an option. In this presentation, I'd like to focus a little bit on the first capturing chromatography step here, the protein A step. And I'd also like to go a little bit deeper into this technology, this continuous chromatography technology. So in batch, this here is our column, and you would start to apply your sample to, let's say, protein A. So the blue color is, let's say, your antibody, and when you apply it to a column, of course, you have a saturation here in the first part, and when some of your product breaks through, you would stop the loading, because you don't want to lose some material. What we do in continuous bioprocessing with the simulated moving MET chromatography is that we just take this slide out of the column and divide it into several columns, 3 to 4, maybe 5 columns, it depends. What we can do now is we can continuously load our product to the column here, and everything that is breaking through can be captured on the second column. When this one here is fully saturated, it goes into the normal cycle. This is wash, elution, regeneration, and equilibration. And the second column moves to the position of the first one and is further, let's say, supplied with or loaded with your protein of interest. And this goes into a circle. So it means you don't lose any material. We are operating close to the static binding capacity, which is the maximum binding capacity you could obtain. And it means afterwards that less resin would be needed, and you can continuously apply or load your product onto these columns. The chemistry and the principle remains the same. So it's still the same sort of chromatography. There is nothing changed, so still protein A, still the same sorbent as before. So what we have done, we have made a case study out of it. So we compared a conventional map purification process coming from a bioreactor with a quite low map concentration below 1 gram per liter. And we measured the operating binding capacity and residence time, so we have a model for that, but I don't want to go further into details here. We had a model, and we compared both. So we compared the batch chromatography versus the SME, the simulated moving bed chromatography. So in case A, we had this batch chromatography, and case B would be, as I said before, as a continuous chromatography, using several columns in a row. So for the batch, we needed 1 column. Operating binding capacity was 26 milligram per milliliter with this protein. And we calculated the specific productivity in grams antibody per liter of sorbent per hour. And we calculated as 3 grams per liter per hour for the batch process. If we now apply this simulating moving bed chromatography to the same process with more or less the same conditions, we are able to increase the specific productivity to 8 grams per liter per hour, which is 2.6-fold, something like that. And as I said before, it's the same material, it's the same protein A sorbent, so nothing changed. But we said, how can we further intensify this process? So it could be, because maybe I should go one back, this here is in gram per liter per hour. It means we can't really change the operating binding capacity. It's a little bit higher because of these breakthrough curves, as I said before, but we can't really change the properties of the sorbent, so it's given by the sorbent. And of course, we can change the amount of sorbent we use, so it's the liter, so the volume of sorbent. But what we also can do is to change the time we need to load, for example, the antibody to the column and all the other steps. And that's what we've done here. So we thought, if we concentrate our load somehow, we are faster in loading it to a column, and therefore the process economics would benefit from it. So we could use, for example, the conventional tangential flow filtration because this can concentrate the load to the column. This is quite easy, but it's not easy to run it continuously. So it might be possible, but there's another way to do it, a simple plug-and-play approach, where we have this device here, we call it Inline concentrator, and we could use it to concentrate, before we load it to the column, we concentrate our clarified cell culture fluid first by a factor of 5.5 or something, and we can apply it then to a column. So the process would look like this here. So we have, again, our load. We have this ILC, the inline concentrator, or SPTF, as a single pass tangential flow filtration module. We would go then to a search tank, but this is optional. We can connect it directly to the column as well. And we can run the same process. As you see here, we have 1 column more. This is to accommodate a certain balance cycle, because now we have to balance all the flow rates and all the volumes. We run through the columns, and that's why we need a 4th column here. But you will see here in the next slide, when I compare it to before. So again, our batch, then the simulated moving bed without pre-concentration, and the combined ILC SMB. You see that the specific productivity, it raised a lot. So it's 30.8 gram per liter per hour now. And that's versus SMB alone is 3.85-fold, and versus batch is more than 10-fold higher. So this is really, I would say, a good option to really increase the specific productivity. You might ask, okay, productivity is one thing, but what about the product quality? What about the process economics? Other questions here. And we have done it again, the same here, and we run the ILC module together coupled with the BioSMB, so simulated moving bed, for more than 20 hours. And we checked for product quality. We checked, is there any change to the product, any change to the run itself? And you can see here that's the aggregate level of the antibody and the HEP level. This is HEP and aggregates over time. And you can see over the time, there are some changes, let's say, to the aggregate level, but it's quite low. So it's in between, let's say, 1.8 and 3.2%, something like that. And it's more or less the same for the HEPs. So it's between 400 and less than 600, maybe 550 ppm. So it's quite constant over time. And also the productivity over time is more or less the same. So we reach the same productivity as in the processes before, which shows that this sort of, let's say, combination of ILC and BioSMB or simulating moving chromatography is stable and does not impact the product quality. Okay, so we have seen it's feasible. We can run it over time. We can run it a longer time, 20 hours and more. But what about the economics? So if it's terribly expensive, you wouldn't take it. That's for sure. And therefore, we calculated using a software. It's called Biosolve software from Biopharm Services in the UK. And we made a cost of good analysis with the same model scenarios, which I've shown before, and with the 4-column process for the capture of 5 clinical batches per year, antibody from a 200-liter bioreactor, and titers between 0.1 gram and 1 gram per liter. Maybe I should say these titers are quite low. So for a batch process, I would say is very low. This is to mimic a continuous, let's say, bioreactor running continuously. That's why these low numbers. And when we compare now the combined ILC, so the inline concentrator SMB versus batch, and again with the bioreactor titer here, you see there is, of course, a benefit, a cost saving over batch, yeah, of 16%, something like that. When we add the ILC here versus SMB, we would have additionally 57% on cost saving. Yeah? And as you can see here, if the bioreactor titer goes or increases, then somehow, somewhen, the ILC combination doesn't make any sense anymore. So it means the blue line, which is the ILC versus batch, it's much better, and there is less benefit, let's say, in combining the ILC together with the SMB. So in conclusion means that if we implement this ILC device, the inline concentrator, into this process, it can increase the capacity, let's say, dramatically. So the productivity goes quite up. But it is a sort of balance. So it means if you get, let's say, 2 gram per liter or 5 gram per liter, it should be a calculation, should be made first to really show if it gives some benefit or not to this. So as I said, the combination of both would lead to a stable process, to a good product quality, and of course to a lot of cost saving in between. So yeah, why not considering to use inline concentration step somewhere in your process? This is not completely stick to, let's say, the first capture step. So you can use it everywhere. For example, for your final concentration. When you have your product is finished, is ready, why not use it there? Just to concentrate it a bit further inline when you elute it from a column somewhere. My manager stated that let's say the most impurity in a process is water. It's just the volume. And by decreasing the volume, you could increase the productivity of your process as well by just implementing a plug-and-play device like an ILC. And that's it from my side. Thank you.
So any questions for Mr. Glantz? No, I think today's my day because I have one. So which other scenarios for the use of this technology could be beneficial for biopharmaceutical development? Other scenarios, other options?
Yeah, for sure. So every time you want to concentrate somehow. So for example, if you scale up your process and you would need bigger containers, for example, somewhere. So why not choosing an ILC just to really concentrate it first? Or if you have several columns, one after one, why not increasing the eluate volume from the first column and decreasing, so concentrating this volume, just to save, maybe container space or whatever, and also to increase the productivity of the next step. So we have seen in certain scenarios, when you go with a concentrated solution onto a column, when you load it, the productivity will rise a little bit. Not only in simulated moving bed chromatography, but in regular chromatography as well, because you are much faster when loading it. Yeah.
Okay, thank you.
Thank you.
Okay, so I don't know if you have any general question for any of our speakers. I think that we didn't have time enough for some questions for Luisa, and your presentation was very interesting, going back to Map Science activities. And I would like to know, okay, me as a CMO also, so we have some natural link between each other, what are the main difficulties or constraints in the development of biosimilars, the manufacturing of biosimilars? If you had to say top 3 difficulties.
I mean, there is monoclonal antibodies. Any high molecular is very difficult to develop and work with, because the modifications can't happen in any step of the process. And many times the most difficult is that you cannot detect that modification. So the challenge, the biggest challenge, is to identify and define your molecule as much as possible in all the possibilities, this orthogonal study of the structure, of the functions, of all these steps. So the development of all the analytical methods that you need to establish exactly what is your molecule and what is the molecule that we want to produce all along the time. It's very, very the challenge, really. And then the second one is to establish the process robust enough to produce your molecule for the life of your product. Because since you are working with live systems, the modifications happens at, I mean, I always like to say, to simplify too much, but at least it's clear that that is like that. We are working with mammalian cells that produce a specific monoclonal antibody. So it's like us, let's say, but in a very small process. So at the end, the modifications happen. And the challenge really is to define our product, to identify if these modifications happens, and to develop the process robust enough to avoid these modifications during the production.
Fully agree with you. Yeah, I've suffered in S3P, sure. Okay, so thank you very much for being here today. Hope you enjoyed this lecture, these speeches, and also you had a nice BIOSPAIN. See you 2020. Thank you.